Nobody was there on yesterday's class. What happened? Did you forget?
Yesterday there was class.
Yeah, it's a medical class for the 11th class. It means for you people. So, anyway, uh, we have to start from the chloroplast. So, first I will explain it. What is the chloroplast? So, day before yesterday, the last class, what I did, let me give you a recap. I told that plastids are the structure which are found only in plant cells. They are not found in the animal cells, right? Plastids are basically of three types. So the first kind of the plastid, they are chromoplast. So the purpose of the chromoplast, they are found in the cell. They provide color to the cell. So in the nutshell, they provide color to the fruits. They provide color to the flower. They provide color to the even leaves other than green color, right? Because green color is because of chloroplast. Then the second one is the chloroplast. So the pigment means chromoplast purpose was the chromoplast is providing the color. Now it's a chloroplast. So chloroplast is of green color and its function is to perform photosynthesis. They provide green color to the plant. So every green part of the plant they are present, right? And their main function is provide the to do the photosynthesis and even the lipid synthesis, formation of the lipid. Then the third one is leucoplast. Leucoplasts are colorless structures, right? They all are plastid, but leucoplasts are colorless. So what is their function purpose? Synthesis of fatty acids, amino acid means synthesis of fat, synthesis of amino acid, and storage of the food. So basic their function is storage of food. They are colorless. They are found in every part where the food is being stored. Then further, these leucoplasts, which are colorless, they are further divided into these categories. So A, elioplast. This is precursor of chlorophyll, right? Then B, amyloplast. Its function is storage of starch and sugar. Elioplast, storage of lipid and oil. Proteoplast, storage of protein. So these are the leucoplasts. Fine. So now we are going to study. So these are the type of the plastid. Now we are going to study chloroplast. So the main function of the chloroplast is photosynthesis. So they are found in every part of the plant where the color is green. Chloroplast is double layered structure, right? So they have double layer, outer membrane and inner membrane. Outer and inner membrane. Inside the outer and inner membrane, so there is a gel-like substance called stroma. So in mitochondria we studied there was a gel-like substance which we call matrix, right? Then try to understand here. So within the gel-like structure, there are these kind of structure, these structure or these piles. So this coin-like structure pile up one on another, right? So these structures are called grana, right? So granum is singular and granum is multiple, means plural structure. So they are called granum, right? So they in a chloroplast, within a chloroplast, there may be 40 to 60 grana. That is an important question for me. Inside a chloroplast, there may be 40 to 60 grana. These grana remains interconnected with each other, right? They are called basically stroma lamellae, these interconnections which connect with each other. So every granum is made up of coin-like structure. These coin-like structure are called thylakoid. So every granum have 2 to 100 thylakoid, right? A granum may have from 2 thylakoid to 100 thylakoid, right? Inside granum, there is a magnesium compound. That magnesium compound is called chlorophyll. So chlorophyll is responsible for photosynthesis. This is the chemical, this is the pigment which is responsible for photosynthesis, right? Then inside the chloroplast, like mitochondria, there is a chloroplast DNA. We call it CLPT DNA in short. So this is chloroplast DNA. It is single, circular, and semi-autonomous, right? And so this is the structure of chloroplast. Look, now let's talk about the photosynthesis. Photosynthesis is a reaction which we divide reaction in two parts, right? 6CO2, 12H2O, right? In presence of light, chlorophyll, it will make C6H12O6, that is called glucose, right? And this is food basically, glucose, right? And then oxygen and water. Try to understand. Fixation of carbon dioxide, because carbon dioxide is in gaseous form. Fixation of carbon dioxide is called dark reaction. it takes place in stroma. And breakage of water, right? Breakage of water molecule is a light reaction that takes place in grana. So dark reaction takes place in dark reaction takes place in stroma, and light reaction takes place in grana. Is that clear or not? Tell me. Is it clear? Elam and Arya?
Yes, sir.
Okay. So do one thing. Please note down the structure of chloroplast and then rest of the thing. Then I will dictate. Note down all this information. You have to CLPT DNA, single, circular, and semi-autonomous, right? This is chloroplast, magnesium compound. This thing you have to note down the extra information which is written over here. Let me know when you note it down.
Done, sir.
Done? Okay. So Martin, done? Alia?
Yes, sir.
Okay. So let's start. Uh, chloroplast possess photosynthetic pigment. Chloroplast possess photosynthetic pigment. Photosynthetic pigment, chlorophyll and carotenoid. Chlorophyll and carotenoid. Full stop. The chloroplast of algae, the chloroplast of algae, other than the green one, are called chromatophores. I have written over here spelling. Are called chromatophores. Next one. The chloroplast may be of various shape. The chloroplast may be of various shape, but In higher plant, chloroplast may be of various shape, but in higher plant, generally of disc shaped, but in higher plant, generally of disc shaped. Full stop. Next point. The chloroplast is covered with double membrane like mitochondria. Double membrane like mitochondria. Full stop. Outer membrane is more permeable. Outer membrane is more permeable than inner membrane. Outer membrane is more permeable than inner membrane. Next point. The ground substance of chloroplast, is called stroma or matrix. Stroma or matrix. Next point. The chloroplast have DNA, which is generally the chloroplast have DNA, which is generally single, naked, and circular, which is generally single, naked, and circular. Next point. Light reaction takes place in grana. Light reaction takes place in grana, and dark in stroma. A chloroplast may have 40 to 60 grana. A chloroplast may have 40 to 60 grana. Full stop. The granum are made up of thylakoid. Granum are made up of thylakoid. Each granum may have 2 to 100 thylakoid. Each granum may have 200 thylakoid. Next point. The function of chloroplast is photosynthesis. The function of chloroplast is photosynthesis. And what else is there? DNA, we discuss it. Diagram is very important. Diagram is very important for your school examination. They can ask you either diagram of mitochondria or chloroplast. These are the favorite questions. So did you note it down this reaction as well?
Yes, sir.
Okay. One more information that algae, the chloroplast, we have already discussed that in plant, that chloroplast of algae are of different shape and they have eye spot or stigma, which is light sensitive. We have already discussed that, right? Pyrenoids are found in algae, chloroplast of algae. Remember or not? Pyrenoids, remember? So this was all about the chloroplast. Any doubt?
No, sir.
Okay, fine. So let's start the another cell organelle. The another next cell organelle is ribosome. Ribosomes are very simple. They do not have any membrane. So ribosomes are basically made up of two different units, right? Try to understand. They are the only cell organelle which are found in plant as well as in animal. So basically, the ribosomes are made up of. They are found in plant, animal. They are found in both prokaryote as well as eukaryote. By the way, prokaryotic cell do not have any cell organelle, but they are the only cell organelle. Means ribosome are the only cell organelle which are found in prokaryotic cell as well. The difference is only that the cell or the ribosomes of prokaryotes are smaller and ribosomes of eukaryote are larger. Right, that is the only difference. Next basic characteristic is that they are made up of two units, a small and large subunit. Right, this is the large subunit and a small subunit. Both subunits remain separated. Right, at the time of protein synthesis, both subunits join each other. Right, so ribosomes are joined; these both units join and they make a proper ribosome. So the bond between these two units means a small subunit and largest. You can say this is a small subunit, this is a larger subunit. Larger fit on the smaller one. So the bond between two subunit is made by the magnesium. So this is called magnesium bridge, right? So both unit of ribosome remains interconnected, adhere or attached with each other with the help of magnesium bridge, right? Then what happened? Try to understand. First, what is the origin of ribosome? This is nucleus. This is nucleus. Inside nucleus, there is a structure called nucleolus. There may normally one nucleolus and there may be more than one nucleolus, right? So normally what happened? Nucleolus is the place where formation of ribosome takes place. That's why nucleolus is called factory of ribosome, right? So inside the nucleolus, right, these ribosome unit are formed. Both of the ribosome unit, they are separately formed. They come out of the nuclear pore, right? This is the nuclear membrane. They come out of the nuclear pore, and after coming out of the nuclear pore, they assemble with each other. After assembly, they make a ribosome. Fine. Now, ribosome is without membrane. It do not have any membrane, right? Assembly takes place in cytoplasm. So ribosomes are produced in nucleus, means nucleoplasm, but their assembly take place means both unit combine with each other in the cytoplasm. Formation takes place in nucleus, inside basically precisely inside nucleus in nucleolus. Ribosome, Robinson and Brown discovered them in plant. Pallade also discovered ribosome, they discovered in animal, right? So both have discovered separately. Now, association of ribosome takes place means both units combine with each other at 0.0001 mole magnesium. At this level of magnesium, they dissociate, and beyond this level, they associate. Ribosomes remains connected with the rough endoplasmic reticulum with the help of protein called ribofourin. So that is a glue-like structure. Ribofourin is a glue-like protein. So ribosome remains adhere with the endoplasmic reticulum with the help of this protein, right? And the unit to measure the ribosome is called Swedberg. So there was a scientist, his name was Swedberg, who discovered a unit to measure the ribosome. That's why this is called Swedberg, right? So Swedberg is a sedimentation coefficient in centrifugation, right? Uh, and then ribosome are found in both prokaryote as well as eukaryote. Then the size of ribosome in prokaryote is 70s. The size of ribosome in eukaryote is 30s, right? And the 70s, they are made up of two subunits. A small subunit is of 30s and large is of 50. So 30, 50, 80. So do not calculate like this way because this is neither a physical unit. This is a centrifugation constant, right? This is something different, right? So it do not follow that your normal rule of mathematics, right? Addition, subtraction, and all that, right? Because this is not a physical unit. This is a type of, this is not a SI unit. It's a centrifugation coefficient. In eukaryote, the size of ribosome is larger than the prokaryote, so their size is 80s. So out of this 80s, a small unit is 40s and large unit is 60s, right? Ribosomes are made up of two things: protein plus rRNA. Protein have positive charge and RNA have negative charge. rRNA here, small r refer for ribosomal RNA. So this is all about the ribosome. Is it clear? I request you to please draw the diagram. Draw this diagram, right? This one, not this one. You can leave this one. Draw this diagram along with this diagram nucleus. Where do they are formed? Please quickly draw this diagram. Then I will dictate for you.
Done, sir.
Done? Okay. Martin, done?
Yes, sir.
Great. So start writing now. Ribosome. Yes, the first point. They are the only cell organelle. First point. They are the only cell organelle found in both prokaryotic as well as prokaryotic as well as eukaryotic cell. Prokaryotic as well as eukaryotic cell. Next point. They do not have any membrane. They do not have any membrane around them. They do not have any membrane around them. Next point. Formation of ribosome takes place. Formation of ribosome takes place in nucleus. Formation of ribosome takes place in nucleus, within nucleus. Within nucleus. Full stop. Ribosome were discovered by Robinson Brown in plant. Ribosome were discovered by Robinson and Brown in plant and by Pallade in animal.
What's the spelling?
This one, Pallade, it is written over here. P-A-L-A-D-E, Pallade, an animal. Full stop. Pallade also coined ribosome term. Pallade also coined ribosome term. Next point. Both subunit, this is important, very important question. Make a star. Both subunit of ribosome, both subunit of ribosomes remains connected with each other. Both subunit of ribosome remains connected with each other with the help of magnesium bridge. Now, please note down this structure: ribosome, and S stand for Swedberg, and Swedberg is sedimentation coefficient in centrifugation, right? And note down this one.
So with the help of magnesium?
Magnesium bridge, with the help of magnesium bridge. By the way, this is the medical question. On what concentration of the magnesium the ribosome, both of the unit of ribosome dissociate? So that concentration is 0.0001. We'll discuss in NEET class, right? So in the meantime, please note down this one: size of ribosome in prokaryote and eukaryote, and size of the small and large subunit. That may be a question in your MCQ. You may get question in your school exam.
Answer.
Done? Okay. Uh, Two more points, please note down. The composition of ribosome, is that they are made up of protein and ribosomal RNA. The composition of ribosome is that they are made up of protein and ribosomal RNA. Full stop. The function of ribosome is protein synthesis. The function of ribosome is protein synthesis. That's why the function of ribosome is protein synthesis. That's why they are called factory of protein. That's why they are called factory of protein.
So the function of ribosome
Is synthesis of protein. That's why they are called factory of protein. Look, so we have discussed in detail about the cell organelle of the cells, means the component of cell, right? They were all component of cell. Now come to the cytoskeleton. What is cytoskeleton? Cytoskeleton is like that framework which give or provide support to cell. Look, assume your room is a cell, right? So that room is a cell, and everything, every part, laptop, that desktop, or everything which is put inside your room, that is a cell organelle. That room is supported by bricks. That room is supported by some small iron rods, small iron rods, right, which is inside these walls, right? And even sometimes these rooms, the roof of the room is supported by some pillar as well. So those things which provide support to this structure of this room, we will call that cytoskeleton. In the same way, suppose that cell have squarish shape, or cell have oval shape, or cell have rectangular shape, whatever the shape of the cell, that shape is supported by some these kind of infrastructure component. They are called cytoskeleton. So cytoskeleton are those structures which are found inside the, look, like you can see. So this is cell membrane. Try to understand. This is cell membrane. This is cell membrane. Below this is cell membrane. Between these cell membrane, these cell membrane, mitochondria is there, nucleus is there, everything is there, right? So they are filled with cytoplasm. Inside cytoplasm, there are some structure which provides support to this cell, right? Which provides framework support to this cell. The number one, microfilament, right? Then microtubules, and there are actin filament. So these microfilament, right? And these microtubules are made up of tubulin protein, right? And they are unbranched and hollow protein, 15 to 25 nanometer. And cilia, flagella, or sensory hair are made up of this microtubules, tubulin protein. Then there are some microfilament structure. This is also made up of protein. And actin fiber, they are purely made up of protein. They are found in both plant and animal, right? So these three are cytoskeleton. So please note down. Put the heading cytoskeleton. Cytoskeleton of cell. Cytoskeleton structure. Cytoskeleton structure occur. Cytoskeleton structure occur only in eukaryotic cell. Only in eukaryotic cell. Next one. Their function is to maintain. Their function is to maintain structure and shape of cell. The structure and shape of cell. Next one. They are of three types. They are of three types. First, microfilament. Second, microtubule. And microfilament, microtubule, and actin fiber, actin filament. Next one. Microfilament, this one. Microfilament are ultra microscopic. Microfilament are means very small. Ultra microscopic. Long, narrow, cylindrical, long, narrow, cylindrical rods. They are made up of protein. They are made up of protein. They constitute 10 to 15% of total cell protein. Okay. Next one. Microtubules are unbranched, hollow, submicroscopic structure. Full stop. They are made up of tubulin protein. They are made up of tubulin protein. Next point. Cilia, flagella, and sensory hair. Cilia, flagella, and sensory hair are made up of cilia, flagella, or sensory hair are made up of microtubules. Their length is unidentified, but having 15 to 25 nanometer, diameter. Next, actin filament are made up of actin protein. Actin filament are made up of actin protein. They are found in both plant as well as animal cells. Full stop. So these cytoskeletons and their function is to provide shape and support to the cell. Clear? Is that clear?
Yes, sir.
Okay. So after the cytoskeleton mode is given, yes. In your NCERT, cilia and flagella. Fine. So next is cilia and flagella. Cilia and flagella is very important. You may get question 2 mark or 3 mark question here, cilia and flagella. The only difference is that, try to understand. Cilia almost they have the both have not almost both have the same structure. This is the structure, right? So flagella, a cell may have flagella. Flagella is longer one, cilia are very smaller, right? Flagella is like tail of any animal, right? Understand? Flagella is like tail of any animal, but cilia is like hair on the skin, right? So flagella, the length of the flagella may be 100 to 200 nanometer, and there may be only 1 to 4 flagella per cell, right? But the length of the cilia is very small, 5 to 20 nanometer, micrometer, but they are numerous, right? And their number of, right? So if we cut down the transverse section of cilia or flagella, you will find that they are made up of these kind of structure. So they have plasma membrane, they have outer membrane. So there is a membrane. Inside the membrane, there are spoke-like structure, right? So they are made up of microtubule, right? So double microtubule, double structure or doublet. We call them doublet, right? So there are 9 doublet. So in doublet form, they are arranged. So in peripheral part, there will be 9 doublet, right? At the center part is called axoneme, right? So outer there is a spoke-like structure. So outer there are 9 doublet, and central part is made up of this is called axoneme. It is made up of 2 microtubules. So we say that in flagella or cilia, the arrangement is 9 plus 2. Clear? So this is the structure. Is that clear or not? Tell me.
Yes, sir.
You have to draw this diagram. Please draw it. This diagram. It's very simple. And note down the length and number of flagella and cilia. Done?
Yes, sir. Done.
Okay. So start writing now. Put the heading flagella and cilia.
Yes, sir.
Flagella are longer. Flagella are longer. 100 to 200 micrometer in length. Flagella are longer. 100 to 200 micrometer in length, but fewer in number. But fewer in number. Full stop. Only 1 to 4 flagella. Only 1 to 4 flagella occur per cell. They are found. Let's stop. They are found in both prokaryotic. They are found in both prokaryotic as well as eukaryotic cell. They are found in both prokaryotic as well as eukaryotic cell. Next one. Cilia are smaller. Cilia are smaller. 5 to 20 micrometer. Cilia are smaller. 5 to 20 micrometer, but numerous in number. But numerous in number. They occur in group. They occur in group. For example, cilia are found in trachea, or windpipe, in trachea or windpipe, trachea or windpipe of humans. Next point. Try to understand first. I will tell you the structure. So look, this is the origination point is called this one basal body or kinetosome, right? They are called basal body or kinetosome. Fine. So they originate from blepharoplast, right? They originate from blepharoplast. So this basal body or kinetosome is also known as the blepharoplast. So this is the root origination point. So the point from where they originate, that is called basal body, kinetosome, or this is also called blepharoplast. Clear? Then this is the origination point. So there is a shaft which help them in rotation, right? And then the structure I have already explained. So please note it down. Both cilia and flagella, are structurally similar. Both cilia and flagella are structurally similar. They possess basal body, shaft, and basal plate. Basal body, shaft, and basal plate. Full stop. Next point. Both originate from basal body or blepharoplast. Full stop. The central or core part, is made up of two microtubules. Central or core part is made up of two microtubules called axonymy. It is written over here, called axonymy. Next point. At peripheral part, 9 doublet. At peripheral part, 9 doublet are found. Full stop. This arrangement is called 9 plus 2 arrangement. Cilia, next point. Cilia and flagella both remains covered with membrane. Next point. The function of both is locomotion and movement. Is that clear? If you have any doubt, please ask me.
Yes, sir.
Clear, all of you? Okay. Alia, what about you? Alia, Martin, tell me please.
Yes, sir.
Chidika, is that clear?
Yes, sir.
Okay, great. Nobody joined us yesterday. I was waiting for you for half an hour. Nobody turned up. So every Wednesday there is a medical class, neat class for 1.5 hours. One class in a week. They'll also give you the revision. Then centriole. Look, try to understand what is the function of centriole. That is very important. Very, very important. So, centriole are this structure is called centriole. Try to understand. This is called centriole. Right? This is structure. These two centriole remains arranged in a cell at 90 degrees. Right? So there is a 90-degree angle between these two centrioles. This is 90 degrees. Right? This 90 degrees. So two centriole remains. Their length may be 0.3 to 0.5 micrometer in length and 0.15 micrometer in diameter. Try to understand. Their transfer section, you will take the arrangement transfer section is almost like flagella we study. So there were doublet, but here there are triplet. Try to understand. There are 9 triplet. At peripheral part there are 9 triplet, but At center, there is no microtubule. So we call it 9 plus 0 arrangement. Here, in what is in flagella? In flagella, you will see there are 9 duplet, 9 plus 2 arrangement. But these 9 are duplet. But here in here in centriole, there are 9 triplet plus 0. At center, there is a no microtubule. They are made up of tubulin protein, right? And they are daughter centriole divide and give rise to the new centriole, right? So what is their function? At the time of, look, this is nucleus, they are present here. At the time of cell division, they move at opposite pole, right? When they move at opposite pole, they start making spindle fiber. This thread-like structure is spindle fiber. With the help of spindle fiber, they pull apart chromosomes, right? So basically, at the time of cell division, right, they pull down the chromosome, right? They pull apart chromosome. So at the time of cell division, centrosome move to the opposite pole and formation of spindle fiber takes place. These spindle fiber keep apart chromosome. So they help in the nuclear division. Tell me, is it clear or not? Is that clear?
Yes, sir.
Draw this thing which is written over here, this diagram, and 9 plus 0, and write down 9 is triplet, and this diagram you need to draw after the class. This pink diagram need to draw after the class, but at least draw this diagram and write it down.
Done, sir.
Done. Okay. So please note down. Centriole are minute submicroscopic. Centrioles are minute submicroscopic microtubular instead of microtubular structure. Uh, they are found in pair. They are found in pair and remains arranged at 90 degree from each other. There is 9 plus 0 arrangement. 9 triplet of 9 triplet of microtubule. 9 triplet of microtubules are found at periphery. 9 triplet of microtubule are found at periphery and center. There is no microtubule, and at center there is no microtubule. Next point. At the time of cell division, they reaches at opposite pole. At the time of cell division, they reaches at opposite pole and pull apart chromosome Full stop. Now come to the nucleus, the most important component of this or most important cell organelle of this complete chapter, and this is the last topic as well. Look, it was discovered by Robert Brown in orchid root. This question is of medical and discovered about time. Even you may get this question in your 1 marker question. So Robert Brown discovered in orchid root. Orchid are a type of plant. Normally there is a 1 nucleus, right? But if a cell have 2 nucleus, we call it dikaryotic cell. And if there is a multiple nucleus in single cell, 1 cell, multiple nucleus, we call it cytoskeleton condition or multinuclear condition. We have already understood this condition in fungi when I was teaching you fungi. We have seen this cytoskeleton condition in different fungi, right? Now, nucleus is called brain of cell, right? Nucleus is made up of, it is covered by 2 membrane, and we call that nuclear envelope. So basically, nuclear membrane is wrong term. Nuclear envelope is formed because that is made up of 2 membrane. Between these 2 membrane, there is a space called perinuclear space, right? Inside nucleus, there is a gel-like substance, and that gel-like substance is called nucleoplasm, right? We call it what? We call it nucleoplasm, right? Then outer membrane bulges out or folded outside, and it make the endoplasmic reticulum. So normally it is there is a concept, and it is considered that endoplasmic reticular are the folding of outer envelope. Then inside the nucleus there are some structure called nucleolus. So nucleolus are called factory of ribosome. We have already discussed that. There may be 1 to 4 nucleolus in a cell. That is an important question, right? Then inside nucleus there is a nucleoplasm, or this is also called karyoplasm. We have already discovered. At some distances there are some spore-like structure called nuclear pore, right? Inside there is a thread-like structure called chromatin fiber and mRNA. Chromatin fiber during cell division it condenses. Try to understand. Chromatin fiber during cell division it condenses, and formation of chromosome takes place, right? Chromosome is made up of two things. The central part is called centromere, and the external part is called chromatin arms, right? And this chromosome is made up of histone, protein plus DNA. Histone is positively charged protein, and DNA is negatively charged structure. That is also important, right? So what is the composition of nucleus? A nucleus is made up of 9 to 12% DNA, 5% RNA, 3% lipid, and basic protein histone 15%, and acidic protein 65%. That is a composition of a nucleus. So I want you to draw this diagram right now, please. And draw as it is, which is visible to you on the screen. Every information, this is rich in the information. So every information is there.
Okay, sir.
Done, sir.
Done? Very good. Done? So quick. Done, everybody? Shidika? Alia?
Yes, sir.
Okay, great. So please start coding now. Nucleus is called brain of cell. Full stop. It is specialized double membrane. It is specialized double membrane protoplasmic structure which contain all genetic information, which contain all genetic information for controlling cell metabolism, and transferring genetic information. Next one. Except sieve cells of plant, sieve tube, remember, except sieve cells of plant and RBC of mammals, except, underline this statement, except sieve tube of plant and RBC of mammal, every eukaryotic cell have a nucleus. Next one. A cell may have single, comma, dikaryotic or multiple nucleus. Next point. In plant cell, it is pushed to, in plant cell, it is pushed to peripheral position due to development of large central vacuole, due to development of large central vacuole. In animal, it is at central position. Next point, please write down the composition of nucleus. DNA, RNA, lipid, basic protein, acid. Please write down composition of nucleus. Note it down.
Yes, sir.
Okay. Fine. The nuclear envelope is made up of two membrane, each of two membrane, each of 60 to 90, two membrane, each of 60 to 19 angstrom thick. Next one. Nucleoplasm is transparent, comma, semi-fluid, transparent, semi-fluid, and colloidal substance which contain chromatin and number of enzyme. Next one. There may be 1 to 4 nucleolus. There may be 1 to 4 nucleolus. There may be 1 to 4 nucleolus. They are called factory of ribosome. They are called factory of ribosome. Next point. Chromatin fiber are made up of DNA and protein. Full stop. At the time of cell division, they condensed. At the time of cell division, they condense to form chromosomes. Full stop. That's it, right? So nucleus, for nucleus, put the heading. Next heading is chromosome. So the last topic which is left is chromosome. It will take 5 to 10 minutes, and we have completed the cell biology, right? And if you have any doubt, please ask. Please ask me if you have any doubt. And please note it down that the chromosome is left, because next class you will forget. By the way, I keep my record, but still. Any doubt? Elam, Alia, Vedika, Martin.
Awesome.
No doubt. So see you guys, and the next class will start with chromosome.